All right, we initiated, we are elongating sort of, but how do we do that? Well, we need a molecule called tRNA. Okay, tRNA is the molecule responsible for carrying and transferring the correct amino acid to build that polypeptide chain. Basically, it brings the amino acid to the ribosome that matches the mRNA so we can build the polypeptide chain, okay? Most of the time we see tRNA molecules drawn kind of like this. Here's your RNA part, if you will, and here's the anticodon. What? Okay, we talked about codons on the mRNA, and I said the codons for the amino acids are on the mRNA a lot of times, right? Well, why? Here's where it gets tricky. Okay, tRNA carries the amino acid, but it carries the amino acid because it matches the code on the mRNA. So it has the anticodon to the mRNA. You need to remember that mRNA has the codon that tells you what amino acid. If you use the anticodon, you will be wrong. Okay? So the mRNA and the tRNA are complements, right, of each other. Think kind of like DNA. And we had our template strand and our complementary strand, right, or our coding strand. Either way they go. So tRNA is this funny looking molecule. It's going to bring the correct amino acid that was coded for on the mRNA molecule that is sitting in that ribosome waiting for it to be elongated, right? Okay. So that leads us to our second step, elongation, okay? The second step of translation, right? New tRNA molecules are going to bring amino acids. They're going to be added together plus a little energy that we take. That's kind of all you really need to know at this point, is that we have to add some energy to form these polypeptide bonds between amino acids. And that happens at the ribosome. Okay, let's look at this picture because I think pictures are the most helpful. Here's our ribosome. Here are two subunits. Notice we have site A, P, and E, okay? So my RNA molecule is going to move through my ribosome this way, and I'm going to build by holding my tRNA here, right? In my P site, that's where I'm going to add them together to make my polypeptide chain. And then at my E site, you'll notice the amino acid stays on this chain here, but my tRNA molecule is going to be empty. Remember, my tRNA has the anticodon that matches, and it's going to release and leave after the amino acid has been added onto your growing polypeptide chain. So it moves kind of down like a conveyor belt would. And we just keep adding to this polypeptide chain at the ribosome until we reach our stop codon. Oh, and the RNA adds the our RNA, right, adds the amino acids as the tRNA brings them in. So it's kind of like a matching piece there too. And then finally, our last step, we reach a stop codon. If we reach a stop codon, that means translation is going to end. We have completed our polypeptide chain, and that polypeptide chain is going to release from the E site. Probably has some more to do, right? It's not really a complete functioning protein yet. It needs to fold, probably even combine with some other proteins in order to become functional. But it releases from my ribosome, my mRNA releases, and even my ribosomal subunits release as well. So that is the end of translation. This is a good image, I think, to look at. Kind of gives you stop codon, start codon. Here's your initiation, elongation, termination, and then how it kind of goes back to that. Good chance for you to go ahead and explain that to yourself. What else should you be able to do? Well, this is the same code we did when we were talking about transcription. We did the DNA code, we did the template code, we did the mRNA, but we did not do the tRNA and the amino acid. I want you to take a moment and pause, figure out the tRNA code, and then go use the codon chart to figure out what amino acid it codes for. This is a good time to go ahead and check yourself, make sure you understand what is happening. Pause the video. In a moment, I'll show you the answer. All right, are you ready for the answer? Here it is. How'd you do? Notice it started with AUG. This would probably actually in real life have to be a longer amino acid code, so there's no stop codon. This was just kind of for practice. And remember, you had your triplets. This was your codon on the mRNA. Your tRNA just had the anticodon, and it brought the correct amino acid. I hope that was helpful, and I hope you understood that.